Hello and good evening to one and all present here. Um, Today I would be discussing with you the week 7 lecture on the course Experimental Biochemistry, which is being taught by Professor Shagota Das Gupta and Professor Shoumode of IIT Kharagpur. I am Srijan Sen, and today I will be taking your live question answer session on this particular week lecture. So firstly, I would be discussing with you a few the slides on the lectures that were taught to you in this particular week in very briefly, and then I would be solving a few assignments that would a few question and answers that will help you in solving your assignments and taking up the future exams that will be coming up anytime. Okay, so let us begin. So I am sharing my slide with you all. Okay. So before we directly jump into the main topic, we discussed about amino acids, we discussed about proteins. Also, we spoke about the separation techniques of proteins and all. So today's lecture will mostly deal with the isolation and the characterization of proteins. So this is a continuation of the last class as well, where we talked about the different types of chromatographic techniques. So this is a continuation of that. So there we talked mostly about the macromolecules or biological macromolecules. Here we would be mostly focusing on proteins. So the last class was just to give you an idea of the different kinds of chromatographic techniques that were there, alongside the different separation techniques that you can use in your daily experiments and research field to carry out the different separation from a mixture of different compounds. Today we would be mostly focusing on proteins. Okay. So the first thing that comes to our mind is how can we isolate proteins? So if we see the different ways of isolating proteins is from natural sources and by the recombinant protein that is expressed from the different sources. So two ways. One is the natural source and the other one is the recombinant source. So if we look from the natural source, we can get proteins from whole organisms, say the entire of a particular organism, say bacteria. That is from an entire organism. Then we have tissues, embryos, tissue culture cells, and different microorganisms. So these are the natural sources from where protein can be isolated. Now, the recombinant proteins that are expressed in the different sources includes your microorganisms, then you have fungi, plants, mammalian cell systems, whole animal production systems, and the insect systems. So there are the two different sources, natural and recombinant sources, and in these two different sources there are several categories from which you can isolate proteins. So the general strategy for purifying your proteins is you take a tissue or an entire microorganism, you disrupt it, then you go through the crude fractionation, and selected fractionation would help you to get your protein. So beside that, you can see the image of a bacteria that has a peptidoglycan layer and there are different organelles present inside it. and inside this bacteria your protein grows. So your protein grows inside this particular bacteria, and what you do, so you take this microorganism, you take this bacteria and you disrupt it. That is, you split it open using different chemicals or heat denaturation. Okay, so you open it up using harsh chemicals. So you break the peptidoglycan layer and you take out all the organelles out of this um, microorganism or bacteria. Now the thing is that your when you break open the cell, what happens? All the organelles are coming out into the solution. Okay. Now you have to isolate only your protein from that mixture. So again, coming to the separation technique, where you have to separate the proteins from a mixture of all the organelles, right? So you have microorganism, you go disruption, then you go for crude fractionation. So crude fractionation is separating the, say, the bigger molecules from the smaller molecules. Okay, so from the crude fractionation method, that is the primary stage of separation, you separate it out. Then you selectively fractionate it, or selectively you separate the different available compounds. Okay, and then you take out your protein of interest. So your protein can be either in the supernatant, or your protein can be there in the pellet. Whatever it is, you can get your protein. So the different ways of protein isolation includes your solubility by salting out, then you can undergo centrifugation, then chromatographic techniques where you have the gel filtration or the molecular sieve chromatography, depending on when you want to separate based on size and mass. If you want to separate based on charge, you have the ion exchange chromatography, then hydrophobicity chromatography, depending on the hydrophobic interactions, and then you have reverse phase chromatography. For binding affinity, you have affinity chromatography and antibodies available. So what is selective precipitation? So it separates based on the ionic strength. Then you have pH, organic solvents, and the molecular crowding agent includes your polyethylene glycol or the PEG. So specific property of target proteins like charge or hydrophobicity, solubility, and size helps you to selectively precipitate out your protein. So when you are increasing the ionic strength of a particular, say, medium, or the protein environment, you are increasing the ionic strength, then your protein precipitates out. Also, when the pH is very close, pH of the buffer is very close to the isoelectric point of your um, protein, your protein precipitates out. Even in the presence of organic solvents, like you have chilled ethanol, your protein will precipitate. So these are the different ways of how you can separate your proteins on the basis of selective precipitation. There are other examples also available. You can definitely check them out. Now see, salt fractionation is another technique. You can see this curve where you have the salting in and the salting out effect. So what do you see? That when salting in, that is in the low concentration of salt, your protein is not soluble. It is soluble at a particular salt concentration. So in this particular region, your protein is soluble, and when you suddenly increase the amount of salt, so what you are doing, you are increasing the ionic strength. So on increasing the ionic strength, your protein just precipitates. So insolubility means precipitating out, or your protein starts appearing in the inclusion bodies, that is in the pellet region. So what does that indicate? That very high salt concentrations that is decrease in solubility as the ionic strength increases, that is salting out effect. On the other hand, mild increase in salt concentration, that is low ionic strength, solubility increases. But if you go very low, that is also the time when your proteins starts precipitating. So this is the curve of salt fractionation, where you can see the two differences between salting in effect and the salting out effect. So the salting in effect occurs when there is very low ionic strength, or your ionic strength has decreased. Salting out happens when your ionic strength has increased. So you increase or you decrease the ionic strength, your protein starts precipitating. So there is an optimal. So you have to do this optimization these optimizations when you are doing research. So only at an optimum salt concentration, at an optimum ionic strength, your protein is soluble, and it shows highest solubility at a particular point or at a particular concentration of salt. So you need to maintain that to keep your salt in the soluble fraction. Now there are some ions that are more effective than others in affecting the protein solubility. So this is the ranking of these particular ions is laid on the Hofmeister series. So you can check the entire series, but the most important ions that play a role is, so you can see that so if you check the Hofmeister series, it is categorized in two parts. One is the positive ions and another one is the negative ions. So what it shows, so the highest salting out effect is for ammonium and the lowest is for guanidinium, whereas the highest denaturing capacity is for thiocyanate and the lowest denaturing capacity is for the sulfate. So higher the denaturation capacity, higher is the salting in. Higher the stabilizing capacity, higher is the salting out. Okay. So when you have a salt like guanidinium, it will just denature your protein and it will increase the amount of solubility of your particular protein because it is denaturing your protein. Okay, so what did I mention? That higher stabilization indicates higher salting out. Higher denaturation means higher salting in. Therefore, there is an optimum salt or optimum ionic strength that you should be using. When we have salts like guanidinium, say guanidinium chloride or guanidinium, any other salts of guanidinium, it will denature the protein. Hello, Freddy. Yeah. So I had started. So in today's class, we are mostly discussing about the isolation of proteins. So just I'm giving you a brief summary of how much we discussed. So firstly, we discussed about the different sources from which proteins can be isolated. You can get them. Then the protein purification techniques, where the general strategy is to get that microorganism, break open the particular microorganism. Like for bacteria, you break open that particular bacteria where you have all these organelles coming out into the solution. Then first, by a crude fractionation, you separate the smaller molecules from the larger molecules, and then depending on the size of your protein, you can selectively isolate the protein from the other organelles. Then the protein isolation methods includes your centrifugation, then the chromatographic techniques, the different chromatographic techniques that we discussed in the last class, and solubility. Okay. So now we were discussing about the salting in and the salting out effect, where the salting in occurs when there is low salt concentration or decreased ionic strength, and salting out occurs where there is higher salt concentration or an increased ionic strength. So in both these salting in and salting out cases, the proteins precipitate. Only in the optimal level at an optimized salt concentration, your protein is in the soluble fraction. So this is the Hofmeister series, where higher stabilization indicates salting out, and higher denaturation of the protein indicates salting in. For salts like guanidinium, it will help to solubilize your protein more by denaturation. So then optimum, or you have to come to a middle point, like you can use salts like um, NaCl, that has negligible effect on the salting in and the salting out thing. So how does it act? The mode of action. So the mode of action is the higher concentration of small, highly charged ions like ammonium sulfate competes with the proteins to interact with the water molecules. So once the water is removed from the protein, solubility decreases, resulting in precipitation. So there is a competition that is always going on, be it for enzymes or for the different ions, and there is always a fight to interact with the water molecules that is present in the surroundings of your particular protein. So in the similar way as the hydrophobic effect happens, so once the water molecules are taken away from the protein molecules, what happens? Your protein starts precipitating. So coming to a very important technique for protein isolation, that is the centrifuge or the centrifugation. So the centrifuge looks more like this. You have an armored chamber. This can be refrigerated, this can be non-refrigerated. So if that is refrigerated, you can use at different temperatures varying from 4 degrees to 25 degrees centigrade, or at room temperature. Okay, then you put your sample over here, and this is the sedimenting material. It spins very high. Then so this is a fixed angle rotor, where this rotor is mostly fixed at a particular angle and it moves. The other type of centrifuge is the swinging arm rotor, where when this machine starts, or the centrifuge just we switch on and we start the cycle, what happens is that the metal buckets starts being at 90 degrees. Okay, so many cell fractionations are done in the swinging arm rotor, so the metal buckets that hold the tubes are free to swing outward as the rotor turns. So you have a cell homogenate over here before centrifugation, and after centrifugation you see the smaller and the less dense components at the top, and the pellet, larger and more dense components at the bottom. So there are different speeds of centrifugation that you can use to isolate the different components. So like I was mentioning about the crude fractionation and the selective fractionation. So first what you do when you just open up the cell having your particular protein of interest, because the bacteria is also growing that particular protein, and you need to take the protein out into the solution. So for that you have different chemicals like sodium dodecyl sulfate. You have harsh chemicals that you can use that will just break open the peptidoglycan layer, and your protein and all the organelles comes out. So then you get the cell homogenate. You collect it and you give so you centrifuge it at low speed. Only the things that you can separate is basically the whole cells, the nuclei and the cytoskeleton that remains in the pellet. The other things remains at the top. Now what do? You discard the pellet and you take the supernet and give it and centrifuge it at a medium speed. When you centrifuge at a medium speed, your pellet contains mitochondria, lysozymes and the peroxisomes. Then again you repeat the same thing. You discard the pellet or you keep the pellet depending on what you require. You centrifuge it at a high speed, and then you get the pellet gets accumulated with the microsomes and small vesicles. And on a final high speed centrifugation you can isolate large macromolecules, ribosomes and the viruses. Okay. So depending on what you require, what are the organelles that you require, or only is it the protein that you require, you can centrifuge it at different speeds. So starting from the whole cell nuclei, cytoskeletons to mitochondria, lysozymes, peroxisomes, then you have the microsomes and small vesicles. Finally, you get rid of ribosomes, viruses, and the large macromolecules. So these are the different speeds that you can use to separate the different components, starting with 1000 G for 10 minutes, then 20,000 G, then you have 80,000 G, and then maybe higher, okay, to get all of these in the pellets. And finally in the main solution you have your protein. So this has been mentioned. You centrifuge at 500 G, then 10,000, then 1 lakh G, and then you get the soluble proteins, right? Good. So in the centrifugation techniques, we will discuss about the ultracentrifugation techniques, that is used mostly for the finer separation and less convective mixing using the density gradients of sucrose and cesium chloride. Then we have the sedimentation techniques that includes your velocity sedimentation and the equilibrium sedimentation. So sedimentation uses the centrifugal force of omega square Rm, where omega is the angular velocity in radians per second, R is the distance from the particular axis, and m is the molar mass of the particle. Then you have the buoyancy force that is acting in the reverse order of minus omega square r m naught, where m naught is the displaced mass. So for calculating the centrifugal force you require only the molar mass of that particle, but for the buoyant force you have the m naught, that is the displaced mass. So you can see FB. And another component that plays a role over here is friction. So the frictional coefficient and the radial velocity, the product of these two values gives you the frictional component. So the direction of sedimentation is on the side of the wall, and in that same direction acts as your centrifugal force, but on the reverse direction is your frictional force and the buoyant force. Okay. So this is your axis, and this is r is the radius from the axis. So size of the protein molecules. So the diameter of a single hemoglobin molecule is 5 nanometer. So the protein molecules can be quite concentrated. For example, the highest physiological concentration of any protein are the crystallines in the eye length that can be greater than 50% protein by weight. You have fibrinogen, that is a large rod-shaped molecule that forms a fibrin blood clot when activated. Okay. So now coming to the recombinant DNA to purify your proteins. So this is a very interesting technique from where you start cloning and then you undergo transformation and finally you get your protein and then you purify it. So for the recombination, what you do is you perform cloning. So what is cloning? You amplify a particular sequence or a particular DNA region that you want to insert in a particular vector or an expression vector, a cloning vector, whatever vector you want, you can put that gene into. So once you clone that particular gene, you restriction digest it using different restriction enzymes and you generate those ends, the overhanging ends, the sticky ends, and you clone it in a particular expression vector. Now using the DNA ligase enzyme and the activity of ligation, you join the sequence that you have amplified, the gene of interest, into that particular vector to get the plasmid. That is known as the recombinant DNA plasmid. Now what do? You take that plasmid, you transform it in a strain of bacteria that is competent enough to take up and grow that particular plasmid, and you plate it in different antibiotic resistant antibiotic plates. So the antibiotic resistant gene is in that particular the vector, and depending on the antibiotic resistance, your plasmids, your bacteria that only take up that particular plasmid will only be able to grow on those particular antibiotic plates. You take those colonies, you grow them, and what so you can see over here you have the plasmid. Those all of these cells grow, you take you grow, you harvest these cells and you proceed for protein expression. Okay. So there are different expression vectors where you have p-dest, pet vector, and then you have eukaryotic expression vectors of yeast, virus, mammalian cells like ped, virus has baculovirus, yeast has ppic. Okay. And there are different host systems, like for prokaryotic we have bacteria, usually we use E. coli, and there are other eukaryotic systems like S. cerevisiae from the yeast. Then there are insect cells, HIC 293, that is the human embryonic kidney cells, the mammalian cell line. So what do? So once you get the colonies, so let's assume you have a plate full of colonies, you give overnight culture from to let the bacteria grow. You inoculate in fresh medium, that is giving a secondary culture. So the first one is a primary culture, the next step is a secondary culture. You check the optical density using cuvette. When the OD comes about 0.4 to 0.8, you add IPTG to induce the protein expression. If you are not using IPTG to induce the protein expression, your protein will not express. So what is the role of IPTG? So IPTG is a molecular mimic of allolactose, a lactose metabolite. IPTG does not take part in metabolic pathway. It triggers the transcription of the LAC operon to induce the protein expression. Now you grow this bacteria for about 12 hours. Say 8 to 12 hours at different temperature, and you harvest the cells. So from the harvested mixture you can get your protein of interest. So it is a very important steps of primary culture, secondary culture, IPTG induction. Now your protein is growing inside the bacteria. You harvest the cells, you break open the cells, your protein comes out, and then via different centrifugation techniques you can get your protein. So what are the different centrifugation techniques or the different purification techniques that you can use? First is differential centrifugation, differential salt precipitation, differential solvent precipitation, preparative electrophoresis, and column chromatography. So differential centrifugation is, just as I mentioned previously, centrifuging at different speeds, at different RPMs. You can separate out the cell organelles from your particular protein. So separation of proteins or any material on the basis of their size, mass, and density. It is a function of the size of the protein and the speed of centrifugation, and it gives us a rough separation. It is basically fraction. The other way of separating the organelles from the proteins is via dialysis. So at this dialysis, you have these cell homogenates. You have a dialysis membrane. Then this is the solvent, and this is the cell homogenate or the concentrated solution. So when you dialyze it, what happens? There is an exchange between the two mediums based on the osmotic pressure. So it allows us to get rid of most of the salt ions, although it is very impossible to remove all the salts completely, because there is an equilibrium from, say, from Z. If it is, even if it is zero, no salt, and even if you have 100 millimolar salt, there will always be an equilibrium that is reached. So you cannot completely get rid of all the salts, but mostly it helps to get rid of all the small molecules. Now, column chromatography is the most common and the best approach to purifying larger amounts of proteins. The highest level of purity and the largest amount of purity is achieved from the chromatographic techniques, and also it requires low effort, and also it does not damage your protein product, and it's the standard method for pharmaceutical industry. We have talked about the gel filtration, ion exchange, and the affinity chromatography. So this is just a brief idea of the different techniques. And if we talk in some in detail, so the size exclusion or the molecular sieve chromatography separates according to the difference in size as they pass through the hydrophilic polymer. So the polymer beads are mostly composed of your cross-linked dextran, which is highly and uniformly porous, like the Swiss cheese. So what happens? The larger molecules comes out first because they cannot fit in these small pores, and the path length is reduced for the larger molecules. On the other hand, the smaller molecules can pass through all of these pores and they get stuck in between these pores, so the path length is enhanced and therefore the time taken by them is more. Therefore, they elute at the last. So for that, you need to have a way to know where your protein is. You have a molecular weight marker. You add a target to your protein. If you want to purify an enzyme, and add a substrate to the tubes and see where does it appear. That is also one way. The other way is that you know the molecular weight of your protein. Let's assume that, and then there are different standards of proteins that are run alongside it that will help you to know that, okay, this is the molecular weight of that particular protein, so my size of my protein is in between, say, 12 and 20 kilodaltons. So that is all about the gel filtration. Apart from this, I had also talked about these chromatographic techniques in details in my previous class. You can definitely check back. So you see, this is the porous polymer beads. This is your so you pour the mixture of your proteins from here is added to the column, and then your protein molecules separate by size. So the larger molecules comes first, the smaller molecules will elute at the end. Then ion exchange chromatography. So you see, the principle is to separate on basis of charge adsorption. It also has the highest resolving power and the loading capacity. It has widespread applications, and it's the most commonly used chromatographic technique for protein purification. So what is that? You charge the beads with different charges. Say if you want to separate a protein that is positively charged, so you charge the column with negatively charged molecules, so all the other negatively charged proteins would just elute out, and only it will bind to the your protein or positively charged proteins. So like here we have charged it with positively charged DAE, DEAE cellulose. Now you pass your protein mixture, only the negatively charged molecules will bind and the positively charged molecules will elute faster. Then you start eluting, your negatively charged molecules will pass down over here. So for so depending on the characteristics of the protein or the charge on the protein, you can separate it based on charging of the columns with the oppositely charged molecules. And it has high resolving power and the loading capacity. Therefore, the applications is also enhanced in this manner. Affinity chromatography, there are different affinity chromatographic techniques that we use on a daily life. One is the nickel chromatography, and then you have biotin, which we use. Okay, so it's based on the absorptive separation in which the molecule to be purified specifically and reversibly binds, that adsorbs to a complementary binding substrate, that is a ligand immobilized on an insoluble support, a matrix or a resin. So what you see, you have the ligand attached to the polymer bead. Your beads are over here. You pass your protein, so the key protein of interest is this one. The protein goes and binds to the ligands. Only your protein of interest will bind to that particular ligand to which it has higher affinity, and then you elute it. So the purification is 1000x better from a single step, and it is the preferred method if possible. Okay, so then you add a solution of another ligand and you elute it. So the protein, you need to keep one thing in mind, is that the protein is not covalently bound to the ligand. So this is about your separation chromatographic techniques that we use for the separation of proteins from a mixture of cell organelles and the cell homogeneous. Okay, we have just 2 more slides before we get into the questions. Great. So another thing is the isoelectric focusing. So all of these techniques have been discussed previously. So isoelectric focusing, as from the name you can understand that it separates proteins based on the isoelectric point of that particular protein. So for example, if you have a buffer of pH 6 and the pi of your protein is say 7, so when the pH of the buffer is less than your pi, your protein is positively charged. So if it is positively charged, it will move towards the cathode, and if it is negatively charged, it will move towards the anode, right? So what happens? So one thing is, by varying the pH, you can focus or you can push your protein molecule towards one direction. That is isoelectric focusing, where we are focusing on the pi of or the isoelectric point of that particular protein. So pH, so keep one thing in mind, pH less than pi means positively charged, pH more than pi means your protein will be negatively charged. So when your protein is negatively charged, it will move towards the anode, and when your protein is positively charged, it will move towards the cathode. So on the basis of that, you can cut these slits into slits and you can separate your protein out of it. The other thing is the polyacrylamide gel electrophoresis. That will also help you to separate the proteins based on their size using the electrical current. And then you have the largest molecules coming at the top and the smallest molecules coming at the bottom. And the other thing is the preparative electrophoresis or the two-dimensional polyacrylamide gel electrophoresis. So it's a combination of the isoelectric focusing and the SDS page. So for example, let us assume that we have a complex mixture of proteins. So first what you do, you separate these proteins based on their isoelectric points. Say at a pH 6, some proteins are 4, have pi 4, some proteins have PIs of 8 and 9. So you can separate based on the pH, and then what you do, you run it uh, in an SDS page, and then they will start isolating. So this is you do it on a horizontal lane. Isoelectric focusing is done on a horizontal lane, and then you do it on a vertical lane. So isoelectric focusing horizontal lane and SDS page on a vertical lane. So first your proteins separate based on their isoelectric points, and then they will separate based on their mass or their size, right? And then you can isolate your protein. Understand, this helps you to give you an idea about the isoelectric point of the protein as well as the molecular weight of the protein. Okay, so that's all about the separation and the characterization of proteins from the natural as well as the recombinant sources. We will discuss with the questions in just a few minutes. Okay. So maybe in 5-10 minutes we can discuss with the questions. If you have any questions, you can definitely ask me from the lectures. From the lectures, if you have any questions specifically, you can ask me. Else you can just wait for a few minutes, and then I would be discussing with the question and answers. You can ask the questions in the chat box. That is great. So let us start with the questions then. I think we can complete early and then, okay, let's see. What do we have? Okay. So you can answer me in the chat box, okay? So we can complete early today. That would be great. Okay. So the first question. In a mixture of four proteins listed below, which should elute third in size exclusion chromatography? So you have four proteins of different molecular weights, 13,000, 68,500, 4,50,000 and 1,45,000. So which should elute in the third place? Just a minute. Yes. So you tell me option B. Yes, that is absolutely correct, because in the first place it is C that is eluting, then we have D, third we have B, and then A. Very good. So question number 2. Which of the following techniques can be used to separate a protein that binds strongly to its substrate? Cation exchange, anion exchange, affinity, and gel filtration. Right? It is mentioned that binds strongly to its substrate, so affinity chromatography. Fantastic. Okay, let us go to the next one. Which of the following techniques can be used to study the secondary structure in a protein? SDS page, circular dichroism, gel filtration, dynamic light scattering. So it is circular dichroism, series spectroscopy. Very good. So from there you can understand the number of alpha helix, beta sheets, the loop region, the percentage of all these structures can be known from CD spectroscopy. Very good. Okay, the next one. In order to lyse cells, sonication is used. This process breaks apart the cells by? This is a very easy question. Yes, it is sound energy. So what do we require? These sonication technique. So when we harvest the cell, so first what you do, you transform, you get the colonies, you give culture, you induce with IPTG, your protein starts growing, you harvest the cells. The next day you harvest the cells in a particular buffer and you store it at -20 degrees. The next day you come, you add lysozyme, a few protease inhibitors, and you mix it very well and sonicate it. So sonication, what it does, it breaks apart the cells by the sounds, and then your proteins comes out into the solution. You give it a very hard spin at 11,000 rpm for 60 to 70 minutes, and in the solution, in the soluble fraction, what you get is your protein. Okay, so that is a beautiful part of it. Okay, so there you require sonication. Imidazole can bind to, nickel, iron, magnesium, sodium. Yes, very good. That is why we have the nickel NTA chromatography, and imidazole helps to elute. Imidazole is required for the elution of proteins from the nickel because it binds to the nickel and your protein just elutes out. So first what happens, you have the NTA that binds to the nickel. Then when your protein is passed through that particular column, the his tags or the histidines bind to the nickel. Then for the elution step, what you do, you add imidazole in higher concentration. So imidazole binds to the nickel, your protein elutes out. Fantastic. So nickel A is the correct option. Next one. Gel filtration chromatography separates proteins based on their surface charge, size and shape, affinity to the chromatographic column, and or PI. Yes, it is according to the size and shape. Very good. Next one. A mixture of three proteins of sizes 70 kilodaltons, 10 kilodaltons, 25 kilodaltons are separated by size exclusion chromatography. What is the order in which they will be eluted? Another easy question. 70 kilodaltons followed by 25, finally 10. All of them come out together. 10 kilodaltons followed by 25 and 70, and 25 followed by 10 and finally 70. You tell me it's A. Yes, A is absolutely the correct answer. The higher molecular weight proteins elutes first, and the low molecular weight proteins elutes at the end. Next question. At 280 nanometer, a protein solution has an absorbance of 7, and the molar extinction coefficient of the protein is 13460 molar inverse centimeter inverse. What is the concentration of the protein? So you get an absorbance of 7 and the molar extinction coefficient is 13,460 molar inverse centimeter inverse. So what is the concentration of the protein? So how do you do it? You know A equals to epsilon Cl. We can neglect the L because L is 1. So C is equal to A divided by E. So that is equal to 7 divided by 13460, and that comes about 0.00052 molar, which is also equals to 0.52 millimolar, right? So this is the correct option. Okay, option B. Get it? Next. Right. So you have a protein sequence that is given above. Which of the following statement for the above protein is or are true? Molecular weight of the protein is 9.5 kilodaltons. The PI of the protein is 10.3 kilodaltons. It will have maximum solubility in a buffer of pH 10, and the extinction coefficient at 280 nanometer is 22.46 millimolar inverse centimeter inverse. Which of the following is correct? First, look at the protein sequence. If you want me, I can just enlarge it. Right. Check it now. What do you think is correct? Let us count the number of molecules, number of amino acids. 1, 2, 3. 6. So you have 20, 40, 60, 70 and 76. So you have 76 amino acids. So let us see. So 76 into 110. How much is it coming? So it is coming about it is coming about 8.36. Okay, so more or less it is 110, but so we have a tool called XPC, where you can put this amino acid sequence and it will give you the exact molecular weight of the protein. But as of now, the molecular weight of the protein is 9.5 kilodaltons, and if you look at the different combination of amino acids that is available, the pi of the protein is 10.3, because it has more of positively charged residues, like you have arginine, then lysines, you have another arginine, then you have a lot of lysines uh, over here, then another arginine. Okay, so the pi of the protein is um, 10.3. And finally, if you check the number of aromatic residues, so you have 1, 2 tryptophans. So what is how many tryptophans? So 1, then you have over here bold, just so it's bold 2, so how many are 1, so let's start with this 1, 2, 3. So you have 3 tryptophans. So 3 tryptophans mean 3 into 5500, that will come about 16500. Okay, and then you have a few phenylalanines and tyrosine residues, so that will add you to give the molar extinction coefficient to be this. Okay, so the molar extinction coefficient is 20.22460 molar inverse centimeter inverse. The pi is 10.3, and the molecular weight is 9.5 kilodaltons. So all the 3 options would be correct, but mostly this question won't be there in your exam, because you have to use the different tools, because more than calculating it verbally or mentally, it's you have softwares that can develop the different characteristics of the proteins when you give the amino acid sequence. Okay, right. So A, B and D is correct. A, B, D, right. Next one. Question 10. The protein in the previous question is in a buffer of pH 7. So this protein is in a buffer of pH 7. It can be purified by. So how can you purify it? anion exchange chromatography, cation exchange, size exclusion, and you say affinity. Okay. See, what did I mention? The PI. PI is 10.3 and the buffer is of pH 7. So your protein is positively charged. Hence, cation exchange chromatography has to be used to purify this protein. Right? So you can use cation exchange chromatography, and so C is wrong. Affinity chromatography using the nickel column cannot be used because we do not have a stretch of histidines. So tandem histidine repeats are not there, so this cannot be used. You can also purify this protein using size exclusion chromatography. Why not? So A and B both are correct. But if it's an MSQ, go for A and B. If it is an MCQ, go for only B. B is the correct option. Because whenever there is a question of pH and the PI is given, and ion exchange chromatography is the better option to go for. Next one, proteins absorb at 280 nanometer due to the presence of which amino acids? Another easy question. And yes, C is correct and right. Very good. Styrosine, tryptophan, and phenylalanine. Okay, very good. Which of the following salts are commonly used for salting out of protein? So you will get that the Hofmeister series in one of my slides. But if you can remember, just give an answer. For salting out, yes, ammonium sulfate. Very good. So option C is used for the salting out. The next question, to determine the size of a protein, which of the following techniques can be used? Which of the following techniques can be used to get the size of the protein? Size exclusion, SDS, analytical ultracentrifuge, or any of the above? No, you can actually use analytical centrifuge as well to get an idea about the size of the proteins. So any of the above options would be true. Just keep this in mind. I haven't discussed about the analytical ultracentrifuge, but that is also a technique where you can separate the molecules based on their size as well, and that will also help you to determine the size of the protein. Okay, so yes, analytical centrifuge is also there in that column. Next, to get an idea about the three-dimensional structure of the protein, which of the technique can you use? Fantastic. X-ray crystallography and NMR spectroscopy. Very good. Both of these techniques can be used to determine the 3D structure of a protein. And finally, this question, a protein without an affinity tag is purified from sonicated crude lysate. Which is the best? Which of the following is the best purification sequence? Nickel column, SEC, ion exchange, salting out, ion exchange, nickel column, SEC, salting out, ion exchange, SEC. It is D. Okay. So let us study the options one by one. So first of all, it mentions without an affinity tag. So automatically the nickel column cannot be used in both the cases. So A and C is wrong. Now the option B is basically wrong because the order or the sequence. So first you do SEC, then ion exchange, then salting out becomes a lot more hectic. So the crude extract obtained after sonication is first salted out. By this, the proteins which precipitate out are removed and separated from the proteins that remain in the soluble fraction. Now then you subject it to an ion exchange chromatography, which can separate some of the contaminant proteins from our protein of interest. And finally, by the size exclusion chromatography, we can separate our protein of interest. Right? So option D is the correct option. Very good. So that's all then from the question answers. Do you have any other questions? If not, thank you then. We will meet again in the next week. Okay. Thank you all.